let's make a quick review over these membrane transports. You all did some lovely skits, I heard, while I was getting my teeth drilled on. Oh yeah, there's a pain in the butt. Oh, pain in the tooth, actually. But you'd have two different pieces, right? Active and passive. The difference between them, please hear me. Passive things go from what concentration to what concentration? Higher to lower. And this is a terrible example. You're going to laugh, I understand. Think of somebody passing gas. Okay, right around you, large concentration. Whew, man. But quickly it spreads, right? Higher to low. Those are all your passive mechanisms, right? Some of them talk about water. Sometimes you need somebody to carry them along. Facilitated diffusion, right? Sometimes water, that's osmosis, filtration going through a membrane, right? But it's always higher to lower. We cool? Then we go to the actives, and now we're talking about active transport is simply a carrier molecule grabbing it, but what direction is it moving? From lower to higher, against the grain, right? Against the flow. It would be like that gas somehow going back inside of you. Really weird, but you won't forget. No, it's a good example. You won't forget. Okay, yes? So a carrier molecule is grabbing on and taking whatever it is it's transporting from a, which way does it go? Lower to higher. Again, opposite of diffusion. Okay? Then you have our pinocytosis and phagocytosis, right? These are all parts of endocytosis. One's doing liquid, one's doing solid, right? Which is which? Oh. These are questions you'll have to know. Pinocytosis is which one? Liquid. Phagocytosis is solid. Okay? You've heard of phagocytes probably. Or phagocytosis is a fairly common word in biology. You're eating, it's eating this other cell. That's how one of the ways our immune system works. Sir? Mm-hmm. Yep. It is. That's a microscopic picture, yeah. Yeah. So this is one type of cancer. This is what he's talking about on page 79. Yeah, and it's zoomed in super much. That's what, 2,250 times zoom? So it's a very zoomed in shot, yeah. Yeah. Are you with me here, friends? And again, I want to make sure you understand these differences, like I'm asking. You need ATP for active transport, you don't for passive. And the reason why is because you're going against the grain, right? Passively, it'll just flow from higher to lower. But if I want to go from lower to higher, I need to actively use energy to get there. Okay? We're good there? All right. So let's dive in a little bit more here to, oops, this is the wrong one, no wonder. Let's talk about this cell cycle. And this is just cells replicating, right? They're growing, they're splitting, they're changing. Two big types. Do you remember? They're on the nice little purple signs in the back of our room here. Meiosis and mitosis. Good. In your group, in just a little pair the person next to you. What's the difference? What's the difference between these two? Quick? Yes, quick. All right, I'm hearing your conversations wind down here. What's the difference between these two things? They're both doing cell replication, right? They're both making copies of cells, right? One cell making new ones. But what's the difference, madam? It does deal with sex cells. You're certainly correct. Those are the ones that in our bodies that do this. Yes, ma'am. Meiosis, they contain half the parent, and while mitosis contains the whole. Sure. So mitosis is making an exact copy, right? Here is a skin cell. Zoom, 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 boom. Here are two exact copies of that skin cell. Are you with me? And we keep going. Whereas meiosis is taking that cell and basically splitting it in half. So you only get half of the DNA material, half of the chromosomal material. Why is that important? Give me a raised hand. Sir? In case what? Too much? Okay, I suppose it could be a way of getting rid of some excess something. Our body doesn't use it quite in that way, but it could be. Yes, ma'am? Does that make sense? In order to make you not an exact clone of your parents, though truthfully you are more like your parents than you possibly know. It's one of the funny things as a teacher. I meet your parents, I'm like, oh, that's where she gets it from. Or that's where he does. Oh, I see. Anyway, it's fun. But you're exactly right. Because when let's go with people, a baby is formed, right? It is half mother, half father. And those two come together to make our new being. Does that make sense? And if I were to try and take two regular cells that have come about by mitosis and put them together, they wouldn't work. Right? It'd be some sort of crazy thing. I need two halves to come together. Okay. Does that make sense? And again, mitosis is how most of our cells work. We're going to spend much more time talking about mitosis. But again, meiosis is a very important part because, we want to have kids and continue on our species, right?
Why do they call it a daughter cell rather than why do they specifically call it a daughter?
I don't know. Probably the person discovered it had a daughter that they liked. And I don't know, truthfully. I don't. The point is just simply that it's an offspring, So I guess you can call it a son cell if you want, but I think daughter's better. Yeah, that's what I would say. Don't tell my son I said that. All right, cell cycle. Moving on. Our cell cycle. Basically, you've got this interphase time. Then you start this mitosis, which we'll spend some specific time talking about, right? The prophase, metaphase, anaphase, telophase. You've heard all this before, I know. And then you have that cytokinesis and then differentiation. And really, all of those except for differentiation, well, I take that back. Interphase is just kind of the cell living its regular normal life, okay? And it does some things in preparation to divide, but it's just living its life. You're doing your normal things. The mitosis and the cytokinesis is when it's actually dividing and separating, becoming new things. And differentiation, we'll talk about that as well, how it becomes, because you realize that all your cells basically start from just one little cell that your mom and dad put together, right? The sperm and the egg come together. And every single thing else in you came from that one little cell. How does it do that? Why isn't your heart where your nose is? Or why isn't your toes where your fingers are? Or your intestines on the ends of your hands? Well, it's weird, right? How does it know that? We'll talk about that. Okay? And this little sentence is kind of an interesting thing. We could spend a whole class period just talking about telomeres. Have you ever taken a copy, like a picture, and put it on a copy machine and made a copy of it? I'm sure you've done that. Have you then taken that picture and copied it again? And take that picture and copy it again? And copy it again and copy it again? Like, what happens to it? Yeah, it starts becoming less clear and it's fuzzy, like it just doesn't look good, right? So this is kind of one of the things they think is happening to our cells. The more it copies, there's still little random errors that come in. And we think these little telomeres, which are the little tips of the chromosomes, are what actually determine how many copies you can make before that cell dies. And one of the really cool research things they're working on is what if we could possibly make it, because these telomeres wear off, like an eraser on your pencil after you use it too long. What if we could possibly make those telomeres not wear off? Could that make you live forever? I don't know, right? But this is one of these kind of research topics they're talking about. It's an interesting thought. Okay. Interphase is basically the cell living its daily life and getting ready to reproduce. So we start that we will actually grow its other structures that we'll need. Because remember, all the mitochondria that we start off with, I've got to make sure both daughter cells have mitochondria, right? All of the endoplasmic reticulum, I've got to make sure both daughter cells have that, right? All the nuclear material, right? The actual chromosomes themselves, they start to replicate and be ready to split apart. That's all happening in this interphase. You don't have to worry about the G1 and G2 and S phase. It's just the preparation. Interphase is living your normal life, so it's in between phases, right? Interphase. And it's getting ready for replication. And then we dive into this. So again, that's basically all of this, right? That interphase spot. And now we start doing, oops, did I go too far? Oh no, I'm okay, sorry. Then we jump into prophase, right? Whereas the actual first phase of the replication. Now, your textbook is very clear about this. It's one of those goofy things. And there's a great picture, if you want to follow along there on page 81. On page 81, it rolls us through this process in a very nice diagrammatic formula. We have these 4 different pieces, but it doesn't necessarily follow it precisely. In other words, we as scientists have labeled these 4 different separate stages, but it's not like the actual cell goes, I'm in prophase now, or I'm in telophase now. No, it's just based upon when we look at the cell under a microscope, we see specific things, and so we label that a certain phase. Does that make sense? But it all just blends together. Yes, ma'am? Right, it's all just preparing itself. Yep, and again, I'm not going to question you on those S and G phases. Per se, that's just in the textbook. But knowing that during interphase, it's preparing. You're living your normal life, the cell is doing its normal stuff, and it's preparing for that replication. Okay, so in that prophase, we have the DNA that starts condensing into these chromosomes. It's in its normal function, normally inside the cell, and it starts to condense, to become smaller, to become tighter, kind of packaged together, right? And the nuclear membrane, remember that's the covering of the nucleus, right? Like the inner sanctum, the most holy place, right? It starts to dissolve that membrane so that way the chromosomes can get out, right? Otherwise they're locked inside this nucleus. We have to let them out. And that's happening in the first piece, which is prophase, right? If you're looking on the diagram, here's our prophase, right? Sorry, there it is. There we go. Interphase here, right? It's getting ready to go. It's completed all the pieces. It's got all the parts ready. You know, like if you're going to build a Lego set and you've put all the pieces out, you've separated them out, you look at the directions, you're ready to go, right? That's what's happening in interphase. So prophase starts. The nuclear envelope breaks down, and you start getting these microtubules formed. You can see the chromosomes. We move from there into metaphase. And what's exciting about metaphase is we can see that the... Sorry, I can't find my clicker here. Thank you. In metaphase, we can see that we actually have those chromosomes all lined up in the middle, right? I suppose I can bring this lovely picture up here, poster, that way you all can see it better. So, which one of these would be metaphase? Right, where they're all lining up down the middle, right? Yeah, all the chromosomes, they're attaching to those spindle fibers on the centromeres, and it's lining up in the middle. Sir?
Aren't there more chromosomes than
Oh, sure, yes. Yeah. 20, 22, 21, 26? Anybody? 23. 23 chromosomes. Yep. Okay, they're all lined up in the center here. Here's our picture again, so you can see it. They're all lined up in the middle, right, in metaphase. Anaphase, what starts happening? They start to pull apart, right? So it's almost like in a PE class when they line everybody up on a line so that you can choose teams, right? Line up, and then you're going that way, you're going this way, you're going that way, you're going that way. And then they separate into their teams. Does that make sense? And we're just pulling these chromosomes basically in half. They look like little bow ties. You pull the bow tie in half, right? That's in anaphase, that they start to separate. And then in telophase, they've been separated, right? The chromosomes are on opposite sides in these two different cells, right? So it starts to, again, encapsulate these chromosomes back inside of a nucleus. So that's what they mean by that nuclear envelope. The outer membrane gets formed again. I'll show you the picture so you can see it again. So here we go. Here's telophase. You notice we're starting to get like two nucleuses, right? Which makes sense because we want to make two cells. So that's our mitosis, is simply focusing on the stuff in the nucleus, right? First, the nucleus breaks down, the chromosomes start unwinding, then they all line up in the middle during metaphase, they split apart in anaphase, and they start to reform the nucleus in telophase. Yes, ma'am?
Is the rounding the nucleolus like the small edge during early interphase?
Say your question again?
Is the, like, in the nucleus, the small like round thing is like the nucleolus?
In the early, I'm sorry, I don't know where you are. Here, yes. 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 Yep. Yes, ma'am?
In the body, how fast is the cycle?
How fast? I don't know if I have an answer to that one. Do you know that one, Patty? Okay. But it would be a reasonably quick time frame. Because again, we're replacing almost every cell in our body on a continual basis. You are not the same person you were a year ago, like literally. It's whole new cells. So, yeah, and it does depend. Some cells do go, like your mouth cells, for example, in your cheeks and in your mouth. Like if you cut your lip or you bite your tongue or something, within a day or two it's healed, right? Very quick healing. But a nerve is super slow. It can take months to heal. Sometimes it never does, depending on what the injury is. Does that make sense? The last bit, you can't forget. So we've got these two nuclei, but great. You can't have a cell with two nucleuses in it, right? So now we have to go to the next part of this cell division, which we call cytokinesis. And again, if you look at your roots, cyto, like cytoplasm, the liquid stuff, right? What does kinesis mean? Does anyone know? Movement. Kinesiology is a study of movement, right? So basically you're saying that the cytoplasm, the liquid, is going to move. That's exactly what's happening. And it's literally just breaking the cell into two pieces, right? Splitting it apart. Excellent. And again, all of the organelles have to go in, right? We can't leave all the mitochondria over in one and none in the other, or we have no energy. We can't leave all the ribosomes in one and none in the other, or that way we can't make any proteins. Does that make sense? Excellent. Any questions with that cell cycle? How you will see this again on your assessment is I'm looking for you to be able, you should be comfortable with the different pieces. So what happens in metaphase? What happens in prophase? I don't like to do that, but I'm telling you that on WAU's test, that's what they have. Like you have to know that in prophase this happens, or in metaphase that happens. It's life, okay? But it's more about understanding, and I get your question, I promise. It's more about making sure you understand the process, right? Nucleus dissolves, the chromosomes line up, they separate, the nuclei come back together. That's your simplest explanation, prophase, metaphase, anaphase, telophase. Yes, ma'am?
So telophase and cytokinesis are pretty much the same?
So the telophase is, they're happening basically at the same time, yes. Telophase is focusing on the nucleus coming back together again. Like it was all apart, so we could divide the cell. Now it's coming back together. That nuclear envelope or membrane is going back around the chromosomes. And the cytokinesis is focusing on the rest of the cell breaking apart and separating. So yeah, they do happen at the same time, basically. Good question. Yes, sir? Continually, over and over and over again. Yeah, sure. Absolutely every day. It's happening. Multiple times a day. It's happening. And again, depending on the cells, of course. Some divide and replicate faster than others, but yeah, very quickly. Other questions there? Good. Excellent. All right, so let's talk briefly about cell differentiation. We're going to roll through this fairly quickly. You guys are just rolling. You know this stuff. So we talked earlier about the fact that cells all start out as one something, right? And they have to specialize. That's what we mean by differentiation. Because the cells in your eyes work differently than the cells in your toes. The cells in your intestines, they work differently than the cells in your brain. Right? But they all started from the same place. So we have to go through this process of differentiation. And that's where it's literally turning on and off different parts of the genes to make it do specific stuff. Does that make sense? Every cell has the instructions to make every other cell, but only certain of them are turned on in that cell. Does that make sense? It's like basically if you think of a team. Okay, let's take a football team, for example, right? Everybody can't play quarterback, right? The team would fall apart. You need one person to be a quarterback, and you've got to have some receivers and some linemen and some running backs. And does that make sense? I apologize for those of you who aren't sports people. But that team, right? Everybody has a specific role, and that's what this is doing, this differentiation. Okay? There are some pieces. When that egg and that sperm come together, there's a great picture. Help me out. Where's that at? It's much better than this. Yes, thank you. On page 82. So those egg and the sperm cells come together to make that, well, it has 1,000 different names. The fertilized egg, the zygote, the gamete. There's literally 100 names for those two cells coming together at different points. Based upon where it is in its developmental process. We'll talk about that. But once they're fertilized, we call them stem cells. Have you all heard of this before? A stem cell? What's so cool about a stem cell? Like it has a stem that comes out like a flower? No. Yes, ma'am. They can become anything they want to. They are undifferentiated cells. Right? They are cells that have the instructions for anything, and nothing is turned on yet. Does that make sense? So a stem cell could become a brain cell, or it could become a heart cell, or it could become a muscle cell, or it could become an intestinal cell. It can be anything. Sir?
What if a cell that's being made like in your foot decides to be a brain cell?
Ah, it starts messing up and doing things it's not supposed to be doing. What do we call that? Sure. That's cancer. Cancer is a cell dividing and specializing in ways it's not supposed to be. We'll talk more about that in a second.
How do they know what they're supposed to be? Like what turns this stuff on?
Wow. That's a deep question. I don't have a specific answer for that, to be honest with you. We know that there are certain chemicals and certain biological processes that cause certain parts of the DNA to be expressed in a certain cell. The actual why, we're still working on that one. Yeah, and some some cells now we know we've uncoded it enough to say, hey, do this, and it'll do it. It'll actually make the kind of cell we want. Others, we're not positive as to why that happens. Yes. Sure, this is good. This is well done. So as an embryo like, right, you've just been born or still being formed, right, in your mother's womb, Your stem cells are crazy unspecialized. Like they can do almost anything, right? Now you have stem cells in your body right now in various places. At the tips of the bottoms of the hairs you have them. In some of your bone places there are stem cells. Those don't necessarily work as well. They can do some variety of things, but they can't totally unspecialize and do anything. So there's some folks who will, like when a baby is born, they'll collect from the umbilical cord these stem cells, and they'll freeze them and keep them, because theoretically, maybe in the future someday, we'll be able to take those embryonic stem cells and grow you a new arm if it cut off. Does that make sense? We're not there yet. We can't possibly do that yet, but doesn't mean you can't pay somebody $100 a month to store your stem cells. Sure, they'll do that. Okay? So once you have the stem cell, now we have a little more specialization. So stem cells are there on page 82. Oh, they do, yes. Oh, yes, they do. Yes. People pay things to do weird stuff. Yeah. Yeah. They do, right. Some people will actually eat, they'll actually eat the placenta. Yeah.
Why?
Well, hang on. Don't be too grossed out now. Shh, stay with me. Remember, the placenta, and if you don't know, that's the sac that covers the baby, right, inside the womb. The baby is born, and TV shows, they end with that. But then you have to actually birth the placenta, too. It's much easier, what I hear. I've not actually done it, okay? But it's much easier because it's a smaller, it's not a big, huge baby, right? But that comes out. So an animal, if you've ever watched any of these Discovery Channel things, the placenta comes out, what do? They break it. They eat it. Because it's a perfect, these animals, right? Because it's a perfect source of protein. You don't want to just, again, in the wild, you don't want to just throw away this easy, good source of nutrients, right?
Like, how do they know that this thing that came out of them they can eat, but then like other things that come out of them?
I think they eat anything. Most of them will eat just about whatever they can. Yeah. The instinct is survival. Yeah, absolutely. So certainly as humans, a lot of folks don't do that, but some do. Again, because here's this amazing source of protein that's there, nutrients. Let's use it. You can get it. You can have it sent away and put into pills, and people will like. So that way, you're not just actually literally chopping up and eating this placenta, but you get these. I imagine. I don't know. All right. Shh. Sorry, we got down that road. My apologies. Shh. All right, come back to me. So stem cells can be anything, right? Stem cells can be anything. When stem cells specialize a little bit, they become what we call progenitor cells. So again, think of this as being the person who is super great at sports. They can do any sport possible, right? But they decide they want to specialize in one sport. Okay? So let's take football. They decided I could do any sport at all. I'm a stem cell, right? I can do anything. You want to play badminton, you want to go swimming, you want to go bicycle jumping, I can do any of that. But now I'm going to focus only on football. But in football, there's lots of different positions, right? There's quarterback, there's offensive lineman, there's receiver, there's running back, there's defensive lineman, there's safety, right? There's all these different things. So I'm a progenitor cell if I'm specializing a little bit, but not perfectly specialized. They could be any of those in a specific area. So again, in your textbook here, a progenitor cell could be, let's say it goes to the integumentary system. It could be a skin cell, or it could be an oil gland, or it could be a sweat cell, or a sweat gland, right? It can be anything in that system. Does that make sense? That's a progenitor cell. And then most of our cells actually go all the way, so they are specialized in just one something. They are just a quarterback. They are just a safety. They are just a lineman. They can't do other things. Does that make sense? Sir?
There's like 5 of those in the picture or whatever. So
Right.
Which one?
The point is that simply a progenitor cell is a little bit more specialized.
So it can be a progenitor cell and specialize a little more, but it'll still be a progenitor
Exactly. Because it could still branch into a couple different things. Yeah. Again, I'm not worried that you understand the whole diagram and label all these different pieces. Just knowing a stem cell can be anything. A progenitor is a little more specialized. And then your final piece is just the actual cells themselves that have specific functions. And exactly. Like in the picture, there's the neuron, there's the skin cell, there's a sebaceous gland, etc.
Do some stem cells just stay stem cells and it's not?
In our body, we really don't have like this central place that had that. It would be really cool if we did. Now, we have used stem cells. How many of you ever heard of Henrietta Lacks? Oh. The Secret Life of Henrietta Lacks is a fantastic book. I would encourage you to check it out. I have it in my library if you want to come get it. About 100 years, no, that's not 100, 65 years ago now, 70 years ago. A lady from Baltimore. She was an African-American lady, had cancer in her ovaries. Actually, it was cervical cancer, my apologies, in her cervix of her uterus. And the doctors took these cells because they were studying this cancer. What they found was these cells were amazing. They would just keep growing. They would reproduce and just keep growing. And you would get perfect copies of these cells. Usually cancer cells do funky stuff, but these cells were fantastic.
So if they got rid of the cells, they would just keep coming back?
Sure, right. She ended up dying from this cancer. She didn't live very long. But these cells they collected from her, they realized, hey, these cells are perfect to like test medicines on and like to see how cells work. So they ended up taking her cells and almost the whole world of genetics and medicine were using these cells to do their testing on. But she didn't know it. Her family didn't know it. Her name was kept out. They called them the Henla cells, like the first two letters of her first name, the last two of her last name. And so they knew that it was these cells, and these were the best cells, everybody used them, but her family didn't know, hey, there's actually still pieces of my mom that people are using, right? And they've been using this for 70-some years. This book tells a story of her family finding out about this, and
Were they mad?
sure, there was anger there. It's a great story. It'll be a movie someday, I can almost guarantee you. It's a fantastic story. But if you're interested, you're looking for a book to read for English class or something, let me know. I have it in my office. The sperm, they just die. Yeah, they just die. Yes? Sorry? The sperm? They're just reabsorbed by the body. Yeah, yeah, or comes out with the regular fluids that come out.
Oh yeah, how do they get the cells? Do they have to like do surgery?
For the hen loss cells you're talking about? Yeah, they actually had to scrape them, take them from the uterus. Yeah. Sir?
There's a movie that came out in April.
Okay.
The Immortal Life of Henrietta Lacks.
There you go. See, I thought I heard something about it. The book is fantastic. I haven't seen the movie, but that's the name of the book, The Immortal Life of Henrietta Lacks. Well, it's a great, great, great book. Highly recommend. Okay, we have no lab today, by the way. This is class meetings. So no lab today. Tomorrow we will be looking at a lab at how cells, the pictures of them, how they work. We'll look at some microscopes. Adios.